and I am a Keynote staff volunteer with the UC Davis Pre-Health Student Alliance. I would like to start by thanking the AAMC for their sponsorship of this year's conference. Dr. Michael West is a gerontologist and stem cell scientist who studies the effects of aging in the field of regenerative medicine. After graduating from the Baylor College of Medicine, he became involved with biotech companies such as Geron Corporation, and thereafter successfully established advanced cell technology. During his time with Geron, Dr. West collaborated with Nobel laureates Carol Greider and James Watson to pioneer research programs in aging and cancer based on the biology of telomeres, based on the biology of telomeres. Dr. West has since become the CEO of BioTime, a biotech company concentrating on the field of regenerative medicine. Under Dr. West's leadership and expertise, BioTime is developing products that are transforming the way scientists and doctors approach age-related regenerative diseases. It is my privilege to present to you Dr. Michael West.
Well, thank you very much. It's a pleasure to be here. As the introduction said, my background has been primarily one of biotechnology. So we talk about translational medicine. What that means is not just the academic pursuit of knowledge, but the translation of that into processes of, the development of products that can actually be sold and used to help people who are sick. As was also mentioned, I'm a gerontologist, and I'm going to give a little plug for those who are thinking about their careers in the healthcare sector to focus in this area, and I'll talk more about that in a minute. I just wanted to say, I was thumbing through this voluminous directory of this meeting, and it made me think, if I had entered the field of medical research, or let's just say biology and healthcare, looking at all of these competing programs and people wanting you to go into dentistry and orthopedics and everything else, I would just be hopelessly confused. And so I'm going to tell you my story, and maybe there's some things to be learned there about how one can pursue a career in science and in technology, and maybe with a little bit more focus than just looking at 1,000 different alternatives. So let me begin, if I can figure out how to advance these slides here. So I think it's been mentioned that it's really helpful to begin a career in medical research with an idea of having a research interest of your own. Many students that I've encountered in the course of higher education, they apply to different programs in different schools. They get into the University of Syracuse or wherever, and then do rotations, and then some lab likes them, they like that lab, and guess what? We work on Drosophila, so if you want to do your career in fruit flies, that's where you'll be. That's one way to do it. That's how a lot of people do it. I think choosing your area of interest is important. At least it was for me. So early on, I got interested in this. And this is the aging process. Why would that be of interest to anybody? Dr. Bruce Alberts earlier talked about choosing an unsolved mystery. After all, if you solve it, you could potentially, have a niche. You could have a thing that you're an expert in. That certainly could be said for what still remains the mystery of human aging. We've learned a lot about how aging works. We know it is terribly important in healthcare. We know it's extremely important and could be because we have a tsunami of aging coming our way with the aging of the baby boom population. So this would meet that criteria of being a big unsolved area of medicine. In particular, there's a little quote under here from an old naturalist who studied basic biology. It pointed out the kind of obvious organic bodies, meaning you and me, are perishable, while life maintains the appearance of immortality in a constant succession of similar individuals. But the individuals are, of course, each dying. And that's a young woman on the left and her mom and her mom's mom and her mom's mom's mom. Something about that picture and that concept attracted me. It was like, I don't know how to explain it. It was like something in my brain was telling me there's something there. How is it that the human species can continually renew itself generation after generation? We're made of a lineage of cells that have been proliferating since the dawn of life on the planet. But we will only live a few decades and die. If you're a cell biologist, how does that work? The question would be, how does that work? And that's what I decided to focus on. And I'm not saying that that's right for everybody. I'm just telling you my life story. So how do you get at that? Well, first let me just show you a little cartoon I found. I actually kind of manipulated this cartoon. So there's an image, and y'all say, I know what I'm looking at. A lot of people that look at aging say, I know what that is. That's wear and tear. I mean, your car grows old, it rusts, so we must kind of rust from the inside out and all that sort of thing. What the picture I showed you earlier of the constant succession of similar individuals with the individuals passing away made me think, yeah, but if we just wore out, why doesn't our reproductive cell lineage just wear out? Why doesn't the human species just wear out? And what you'll see oftentimes in biology is that the reality is a surprise. There's something really amazing generating the image that we see in aging. One of my clues that this was going on with aging, and that there were actually mechanisms in biology, is Dr. Albert showed us all the DNA polymerase and all that, the cool machinery that replicated the DNA. The evidence that there's a machinery of aging can be seen here. This is an experiment that nature has performed in a sense, not that the scientists have done. On the left is a syndrome called Werner syndrome, and it's a premature aging disease. So on the left you can see a young Werner syndrome patient, and that same patient at the age of 48 years, when she still should be relatively young, and she has gray hair, and one of her eyes had to be removed, and has typically, severe coronary disease and other aspects of aging. On the right, you see the boy, not the cat, has progeria. It's called Hutchinson-Gilford syndrome. And these children, you may have seen them in magazines, they grow old and die in their teens. They'll usually die of heart disease. And it's not because they ate too many steaks and they had a bad lifestyle. There's a genetic mechanism here. How does that work? But what this made me think was, if we could, when I entered the field in the late 70s, we were just getting all these abilities to manipulate DNA and study DNA. If we could get at the DNA mechanisms of those diseases, it would teach us something about aging. And aging is the #1 killer. It's killing everybody. But it's also age-related degenerative diseases are underlying some of the largest unsolved and most expensive healthcare problems for our country. So where do you begin? Well, I began with this. So my first day as a graduate student, I chose to be in an aging research lab, and Dr. Sam Goldstein's lab, and I sat down in a microscope and looked at a picture similar to the one on the right. If you grow cells in the laboratory, they'll replicate and they age. So if you take skin cells from a human and let them replicate, they'd replicate and divide and divide and divide, and then about 50 or 70 doublings, they stop. And the cells on the right are old and they've stopped dividing. And I remember looking through a microscope at these cells asking, what is it in you, the cell? I didn't expect the cell to answer me back. What is it that's causing these cells to age? What does it look like? And I got to say, it didn't look like anything except a big, fat cell that wasn't dividing. So where do you begin? Well, I'm not going to take you through the whole history of this, but in my postdoc work at UT Southwestern Dallas, I had come to the conclusion that the clocking mechanism was at the ends of the DNA strand. So it was in the DNA. You see those little lit up areas at the ends of the chromosomes? Those are called telomeres. And the idea is that like a fuse is a clocking mechanism, or a candle burning, let's say. It's going to look like candle wicks burning. As the telomere shortens in our body cells, those telomeres would shorten, and that would be a clock, and that was the cause, that was the clocking mechanism of cellular aging. And then the idea would be that there was some kind of gene that could reset that telomere length, like a key that winds an old antique clock, and that gene that could reset the clock could make cells not age. And that was why our reproductive cells keep making babies forever, is they have a gene called telomerase. And so my original picture, humans keep making humans forever, is because there's a lineage of cells that can maintain telomeres, and that makes cells immortal, and that cells in our body don't have that, and so the individual passes away. Does that make sense? Way out on a limb. In academic research, you could build your career, probably get grant support. Might be competitive, might be tough, but you could build a career in this area. I looked at this and I thought, you know what? Maybe I had a little bit of a business instinct, maybe. But also I thought, you know, if I spent my whole career in academia, I might have a lab and, 10, 15 people and compete for grant support and so on. But if I could do a biotech company, I could probably have 100 plus people working. And this whole telomere thing is going to take a lot of money. And so I went out and talked to some venture capitalists. And here's an early slide of the early days of a first company I started called Geron. Geron means old person in Greek. And this is an early slide. So you can see we were only 41 people at that point. We were proud of being 41, not 40, it was very early days. We had attracted some prestigious scientists like Jim Watson, who had a real interest in the biology of aging. Len Hayflick, shown there, was the guy who discovered that cells age. So we had this really cool group of scientists collaborating with us, and we had some of the best venture capitalists in the world. Kleiner Perkins, that you see there, was the venture capitalist that did Google and many other famous companies. So off we went to the races. When we looked at these telomeres in cells that age, you can see on the left part here, mortal cells lose, you can see the candle wick burning, the clock was ticking away. And then when cells immortalized, you can see that telomeres stabilize, and now cells were immortal. The question was, could we get telomerase from this immortal lineage of cells that connects the generations and transfer it to cells in the body? Well, that required that we get the gene. And how do you find this gene? I think in academia we wouldn't have found it in time anyway, because we put, I think, about $40 million in this biotech company into being the first to get the genes isolated and to test them. And we beat MIT by 6 days. So we got the patents at Geron and everything, but it was a huge effort. And when we dropped this gene, actually a fun story, Len Hayflick the guy who discovered that cells age, came by my company one day and gave me a little piece of his leg, snipped a piece of skin. I know it's a crazy story. Handed it to me, and that is his skin on the left, and there is Len Hayflick's own cells growing out of it. And it was joking would measure the true Hayflick limit, how many doublings Len Hayflick's cells would go. But what we did is, because we had his cells growing, Len's personal cells were the first cells ever to see the telomerase gene artificially introduced. It was kind of a fun little anecdote. And it worked. I'll be darned. One gene found by first these clues in progeria and Werner syndrome, and then, a biotech company and a lot of money and a lot of great scientists working very hard. We put telomerase into cells, and it alone is capable of immortalizing human cells. Now, what this led to, and now I'm going to quickly race you through an example of how amazing things can happen with some of these just core discoveries. What this led to was an understanding that there could be potentially some cells in that immortal lineage of cells that connects to generations that we could isolate and use in medicine, and they're called embryonic stem cells. And so at Geron in the mid-90s, I organized an effort out of there to isolate these cells from these IVF embryos, left over or thrown away, fertilized egg cells. And on the inside, that little cluster of cells on the inside of the circle called the inner cell mass, the theory was, based on mouse work, that if we could isolate these cells and expand them, we could, and you see there a colony of cells, that blob is a colony of about 1,000 cells. Were one of the first lines President Bush talked to our country about back in 2001 of human embryonic stem cells. And we captured those cells and realized they could be, because they were an immortal cell, could be an infinitely renewable source of all the cells of the human body. And that gave birth to the term regenerative medicine. We realized now we had a way of manufacturing on an industrial scale and manner all of the cellular building blocks of the human body. And there are lots of them. Can you see where I'm going with this? There is a lot of room for the future of medical. Don't think everything's been discovered. There are thousands of lineages of cells that are born from these pluripotent stem cells. And as a young researcher, you could put your name on, well, not just one, but whole big branches of this, because most of these cells are not yet characterized that branch out of embryonic stem cells. And quickly, to show you how such groundbreaking endeavors can lead you into unexpected areas, the embryonic stem cell thing became, as many of you know, a very politically charged field, and then nothing was more politically charged or controversial than the use of nuclear transfer in such biology. So what is that? Well, if you take a cell from the body, the gray cells there, the somatic cells, and put it back into the germ-line, back into an egg cell, you can clone an animal. The guy who first came up with that idea was a dear friend of mine named Keith Campbell, and there he is on the left. He's a really, really cool guy, no longer with us. And Keith, everyone said you couldn't do this, and he did it anyway. A lot of fun stories I could tell you about that. And he cloned Dolly, the sheep. She was cloned from a breast epithelial cell, and so they named her after Dolly Parton, which is a terribly stupid thing to do. Now, what we showed then was the nuclear transfer, and here with cattle, you can see at the top, this is some of the work my colleagues and I did at ACT. If you took senescent cells and put them, old cells, and put them back into an egg cell, the cloned animal would be born young, and so telomeres actually got reset in the process. And so what we realized was we could potentially take a cell from a patient and make these pluripotent young stem cells that could be used to make all the cells of the human body. On the right are the first human nuclear transfer embryos, and this is an example of where the pursuit of scientific and medical technology and knowledge can lead you into, unexpected areas of political controversy and debate, and certainly the first cloned human embryos generated just that. Today, we now can reset telomere length. On the left there, you can see an example of resetting telomere length and reprogramming just using genes. So the controversies are now long forgotten, and take cells sort of in a time machine back to the beginning of life. And many of these technologies I've walked you through, that telomeres and this regenerative medicine revolution, are now in the clinic. And so here is a patient with a spinal cord injury being injected with cells made from H1, the first human embryonic stem cell line ever made. Still, remember, these are immortal cells. Those cells can be making product forever. And these cells have been demonstrated safe, and we're now entering clinical trials to see if we can demonstrate efficacy or usefulness in treating disease. Here are some cells for treating blindness that we've recently requested the FDA to allow us to begin clinical trials on. So I told you a little bit of a history, how, having a focus, maybe focusing on aging is a good idea. Biotech is a different way, a different path than academia. I want to talk just quickly about in my experience, some of the fields that have really expansive potential, where biotech companies will be hiring people and academia will be hiring people. One example is in understanding all of these thousands of cells that originate from pluripotent stem cells and in other areas of medical research. Bioinformatics is a big area. What I'm showing you here is about 200 different kinds of cells that we made from pluripotent stem cells, and then using microarray genes, we look at the expression levels of some 30 to 40,000 genes in these cells. And this is just an abbreviated, what's called a heat map. Each of these little dots is a gene in a particular cell type. And this gets really complicated, really fast. And so, our company needs, other companies need, academics need people who really are good at computer science and dealing with big data. And coming back to whoever's doing the research and saying, here's the one gene or the few genes I found that are really interesting out of this sea of millions of data points. Another area, which is a growth area, is understanding the gene expression and all of this in mammalian development. So here's an embryonic mouse, and some of the genes that we look at, and we see some of these cells expressing these genes, and we ask the question, where in the mouse is this expressed? So correlating real biology with our stem cells, and then for medical research, these are areas that are huge growth areas, and careers can be built. We started the first database mapping out genes, cells, and diseases online. You can find them online in the Life Map Sciences databases. But if you look at this, this is really like primitive maps of the United States where, it maybe has Florida in the wrong place. When we first colonized the Americas, when Western cultures were colonizing the Americas, it wasn't a very good map. Today we have, GPS and much more sophisticated maps. This map of human biology of genes, cells, and diseases is still very rudimentary, and there's a tremendous opportunity for growth. Another area that's expansively growing, because the pluripotent stem cell is all-powerful and can make every cell in the human body, and it can replicate without limit, it opens the door for gene targeting. meaning like a word processing program, like let's say Microsoft Word, you can search and replace a word in a big text. Now you can go into these cells and change the DNA in any way you want and make any kind of human cell and engineered in any way you want. This gives mankind the ability to do amazing things with human cells for medical research purposes. And this area is just beginning. I mean, it's like the day that Christopher Columbus got off the boat, or whoever got off the boat first. It's just the very early days here, what is possible to do. There is the area of personal genomics. So now the cost of sequencing patients' DNA, or just a normal healthy person's DNA, is less than $1,000. That's Moore's law on the halving of the cost of making a number of transistors, I guess, doubling the number of transistors on a chip or something. The cost of sequencing the genome has been decreasing even faster than Moore's law would predict. And all of that data needs to be analyzed and incorporated into medicine, especially in an era, now here's my pitch on gerontology, especially in an era when we have a tsunami of healthcare costs coming to the United States. The baby boomers from World War II generation are now coming to the shores of the clinic, and this is going to bust the bank of the United States. Trillions of dollars of unfunded healthcare costs are coming to our shores. Many of these diseases, in my opinion, could be addressed in novel ways to cut the costs of chronic, debilitating, degenerative diseases and cut the costs to society. And a wonderful area where not only a career could be built, but you could benefit peoples of the world. I guess part of my vision, too, is if you bring all this together, computer science, genomics, the problems of aging, it's possible now on supercomputers to model lifestyle things, genomics, medical histories, and with these Bayesian network analysis algorithms that supercomputers can do, find patterns in human health that are predictive and give insight into the origins of disease and help us improve the quality of medicine in ways that the human brain simply can't do, that can be done with these supercomputer algorithms. So in summary, I would say that we're entering an era that far surpass what we had in years past for deciphering disease. And if we could figure out some of these things like the telomere hypothesis with such primitive tools we had just 15, 20 years ago, could you imagine what could be done in the coming decades? We have, as I said, the ability to now do gene targeting, so it's already in play. We could make any kind of human cell genetically modified in any way, driven by big data, by these supercomputer algorithms that predict the associations of genes and cells with diseases. So, really, the irony is, in an era when there's, so few young people entering the field of healthcare and medical research, we're in an era that the opportunities are so expansive compared to what they've been in the past. It's really ironic, and when there's such great needs with an aging population. So I'd make that one last pitch. I think that cell senescence or cell aging has finally come of age, and I think it's a great growth area for the future for you to consider. Thank you very much. I think it's about lunchtime, but if any of you have a question, I'd be happy to address it. Yeah. How would I suggest the person, I'm sorry, what was, I heard 90% of it, but just not the 10% I needed to hear. Yell it at me.
How would you suggest, you talked about doing rotations, but not I mean, you talked kind of badly about rotations.
Well,
but you-
I wasn't trying to be
Yeah.
critical of rotations. Is critical. How would I do it instead?
Yeah.
Well, yeah, I mean, I guess it depends on where you are in your career. I mean, if you're already in a school and, you don't want to leave that school necessarily to pursue. But if you haven't yet applied to the school, what I did, I mean, and this is just one way of doing it. What I did is I was interested in this biology of aging. I saw a TV program on it was talking about progeria, this premature aging syndrome, and I said, there's biology there. And I sought out the one lab in the world that was doing this sort of thing. And so I went and met with the professor, and he walked me into the admissions office and said, I've got this young guy and I like him and I'd like him to come work, and he wants to apply for the graduate program. And that was a good introduction. I mean, as a random letter applying to a program. That's how I did it. I'm not sure that that's normal or the right way for everyone. But if you have not yet applied for a program at a particular university and you want to do graduate research, I would encourage people to have a sense of what fascinates them, what kind of an area. This is your life's work, maybe, and then finding the schools. In terms of maybe a good path there would be if you say, well, who's strong in, let's say you're interested in fruit flies or something and you wanted to study the genomics of fruit flies, this is an example, as applying it to medicine. One way maybe I would recommend is calling, just look on the internet and find some professors who are expert in that particular area and say, listen, I'm thinking of a career in this area, and could you take 5 minutes to give me some recommendations of where the strongest programs are? And I think most people will be happy to point you in some directions. If you're in a school, I think the rotations and picking the department that best fits your interest is the best way to go. I'm not an expert in academia. I've been in biotech my whole career. Any other questions? Yeah.
In your work, have you been able to discover or paid heed to the relationship between cellular senescence and atherosclerosis?
Yeah, of course. There's a great interest there. There's clearly a profound link, but it's a bit of a mystery. Early on in my career, so for those of you who don't know, atherosclerosis is one of the serious problems of aging, and is what generally is the cause of the demise of these children with progeria. And so historically, people say, well, it's the accumulation of these lipids in your arteries that feed the heart muscle, and so they get plugged up with this fat, from too many greasy hamburgers or whatever, not picking on the hamburger business. Clearly it's not that. That's why I made that little point that children with progeria haven't been misbehaving themselves in terms of their diet. The clock of cell aging was set incorrectly, in a sense, with progeria. So the question is, what cells in the coronary artery could be aging? Is it the endothelium on the inside wall of the vessel? Is it the smooth muscle cells that surround that? Or is it even maybe the perivascular cells themselves? I think most people would point to one of the three. Few people have thought about cell aging and coronary disease, strangely enough. There's evidence, old evidence, that the endothelium itself is senescent in co-localizing with the plaques. There's newer data just coming out in the last few years that there are changes in perivascular fat that's playing an important role in the disease. We measured telomeres early on in the smooth muscle cells, and they appeared senescent as well. So bottom line is I don't know the answer, but most people would, to think about cell aging and atherosclerosis, think of those three cell types and maybe some combination of all three. take a whole day talking about that. I'm happy to talk to you afterwards. Is that it? Thank you very much.